All right, guys, we are going to finish up the chapter on cell membrane and cell transport. So we started talking about active transport last time, and if you remember, it was low to high concentration instead of high to low like all the other ones that were passive. Now, the big thing with active, it did require energy. And then we started talking about different things that do require energy in the cell, meaning endocytosis and exocytosis. Now, for endocytosis, we have two different forms we're going to go over. It's pinocytosis and phagocytosis. Pinocytosis is cell drinking. That's when the cell takes in fluids mostly. The other one is phagocytosis, where the cell is taking in large particles or even whole cells. Well, why would the cell take in whole cells? Maybe they are going and being transferred over to the lysosome, and the lysosome is breaking them apart. Okay, so that'd be a reason for phagocytosis. Now, when we do this, we do need energy in order to go through pinocytosis and phagocytosis. All right. Another thing for digesting whole cells, if there's a virus or a bad bacteria, the cell will take that in, and again, that would also be digested by the lysosomes. Opposite of this is going to be exocytosis. So we are going to take something out of the cell. This was when we formed the protein and the vesicle expelled whatever was inside, in this case the protein, from the cell. Another thing would be toxins. If there's any toxins inside the cell, we are taking it, fusing that vesicle with the cell membrane and pushing it out. Okay. Here is a picture of exocytosis. All right. So we got the vesicle fusing with the cell membrane and then the protein leaving. After that, that vesicle would become part of our cell membrane. Right. Again, we would need energy for this to happen, okay? So here's an example of active transport, guys. We're going from low to high concentration here, and we need ATP, all right? Sorry, guys, picture's a little blurry, but these solutes be much too big to pass through the cell membrane, okay? If the solutes are too big to pass through our cell membrane, we have to move, instead of the solute, the solvent, okay? If we are moving that solvent, we are going to be going from wherever there is less solute to more solute, okay? So in this example, we'd be moving water instead, and the water would leave the cell and go out. So this would be osmosis. If you want to be specific, we have a hypertonic solution and a hypotonic cell. How about this one? We got a vesicle, and it looks like on this side we are bringing it in. Take a solute particle, whatever it might be, we're bringing it in. So this would be endocytosis. Probably pinocytosis. Reason being is it's just one little solute particle, okay? There's probably some water in there with it. All right, this one. We're taking in glucose through our carrier protein, and it's going from the outside of the cell to the inside cytoplasm. This one is going to be facilitated diffusion. This one, so we're moving small, non-polar things. I want to show you guys this picture, but small non-polar things, oxygen and carbon dioxide. Okay, there's carbon dioxide, oxygen, small non-polar. They can move right through the phospholipid. I found this picture online, and it's actually wrong because water cannot move right through the phospholipids. If you remember, water needs an aquaporin to go through. So these two would be diffusion here, but this third one would not be regular, simple diffusion. Right. Here's a fun picture. You can see the little green arrows there. We're moving something very big out of the cell, all right? So that is going to be exocytosis, probably a phagocytosis because it looks like they are very large there. Yeah. Once again, guys, solutes are much too big. What is hypertonic? What is hypotonic? Well, the inside of the cell is hypertonic. The solution surrounding the cell is hypotonic. This means that water would be moving into the cell wherever there is more solvent. So that's where our water would go. All right. With this, our cell would get much bigger and possibly go through cytolysis and burst. This one, guys, we're moving from high to low. We don't need ATP. So we're moving high to low. We don't need ATP. We're going through a protein, so this would be facilitated diffusion. Right? Another one, guys, looks like we're moving in a very large vesicle. So this all would be endocytosis. Okay, the first one's probably phago. These last two are probably penocytosis. All right. Here's another one of diffusion, guys, moving right through the phospholipids. And that is it. Okay. So that's the end of chapter 5, guys. Make sure you know the difference between all of your different versions of cell transport, whether it's diffusion, osmosis, facilitated diffusion, active transport, endocytosis, exocytosis. And endocytosis is grouped into penocytosis and phagocytosis. And osmosis is grouped into hypertonic, hypotonic, isotonic, whether it's the solution or the cell. Okay, so make sure you know the difference between all of them. Make sure you know the mechanisms behind all of them. Make sure you can identify it based on a picture like we just did. All right, guys, have a good rest of your day.